Nerd Globe, Thinking Out of the Box, is a competition held for the students to come up with their queries. Each of you will be provided with chits to jot down questions after each talk. You can put them in the box outside the hall during the break. The best questions will be selected for the student interactive session tomorrow. So the first speaker for the day is Dr. Victor Nizet. Dr. Victor Nizet is a professor and vice chair for basic research and chief of the Division of Host Microbe Systems and Therapeutics at the University of California. Dr. Nizet received his medical training at Stanford University School of Medicine and completed residency and chief residency in pediatrics at Harvard University's Children's Hospital in Boston. Dr. Nizet leads a large basic and translational research laboratory focused on discovering virulence factors of gram-positive bacterial pathogens, elucidating mechanisms of host innate immunity, and novel approaches to infectious disease therapy. Dr. Nizet has authored over 340 peer-reviewed publications, including several in the most prestigious scientific journals. He is the recipient of several awards like the American Heart Association Career Investigator Award, the American Asthma Foundation Senior Investigator Award, and many more. Sir, we welcome you to deliver the keynote address.
So thank you again, everyone, and I want to continue on the themes that I introduced during the inaugural session and challenge us all to redefine antibiotics and think outside the box from the current paradigms to inspire creative solutions to the antibiotic resistance crisis. So, as I mentioned, my laboratory studies the interaction between common bacterial pathogens and the innate immune system. And in general, our innate immune system is quite a formidable adversary. Oops, I actually have control over the direction. We are colonized with potential bacterial pathogens on and off throughout our lives without getting disease because the innate immune system is able to restrict them to the skin and the mucosal surfaces where they cause no harm. But occasionally, bacterial pathogens are able to spread deeper in the body to cause medical problems. And when they do so, we recognize them as pathogens and we determine that they are expressing virulence phenotypes. They have virulence factors that distinguish them from all the good citizen bacteria on our bodies that never cause disease. And our lab has been interested in the discovery and characterization of bacterial virulence factors, but also in a greater understanding of the innate immune system, why it works so well the majority of the time, but why it breaks down, allowing pathogens to spread somewhere. And I think the clues to the antibiotic resistance crisis lie in understanding this dynamic from both sides. So over the years, our laboratory has touched on many pathogenic microorganisms. In particular, we've been interested in the invasive streptococcal pathogens, group A and group B streptococcus, Staphylococcus aureus, which is the leading cause of infections in the communities and hospitals in our country and many other countries in the world. Pneumococcus, which may be a leading killer among bacterial pathogens, producing pneumonia and sepsis and meningitis, especially at the extremes of age. And because of the antibiotic resistance crisis that was highlighted with the NDM1 strains and colistin resistance among leading gram-negative pathogens such as Pseudomonas, Acinetobacter, Klebsiella, our lab has begun to study those in detail as well. And from the host point of view, we pay equal attention to the functions of the innate immune system, endogenous antibiotic molecules that our body makes as a first line of defense against infection, white blood cells such as neutrophils and macrophages, and the way that they are able to survey the body, capture and kill microbes. We're very interested in transcriptional regulation, receptor signaling, cell death pathway regulation, and often study the glycobiology of the host-pathogen interface. All our cells are covered with sugars. The bacteria are covered with sugars. Sugars are a little difficult to study, and people have ignored them compared to gene-encoded proteins, but there's a lot of interesting discoveries to be made there. So the antibiotic resistance crisis is of global concern. And you can see that my country of the United States and India are not immune from high-level antibiotic resistance problems. The genesis is maybe a little bit different in both countries, which have their unique epidemiology and most prevalent bacterial species. But it's a huge problem, but it's not a mandatory problem. There are countries such as Sweden, where my wife is from, or the Netherlands, where I have many collaborators, that have very low antibiotic resistance rates due to much better restriction of overprescription of antibiotics, better hospital epidemiology, restriction of their use in agricultural settings. So it is a public health failure in addition to the biological factors. This is a patient whose name is Bryce that I took care of as a pediatric infectious disease physician in our children's hospital in San Diego, who was doing just great and celebrated Christmas with his first visit to Santa Claus. But a few weeks later, having never been to a hospital or healthcare setting, he developed a cold-like infection that developed very rapidly to pneumonia, empyema, infection of the lining of the lung, and then sepsis. And this was with a community-acquired methicillin-resistant Staphylococcus aureus strain. And he required mechanical ventilation, multiple chest tubes, and ultimately survived the infection. And I'm able to show you his picture because Bryce and his family have become advocates for the Infectious Disease Society of America's Bad Bugs, No Drugs campaign to tell his story to increase understanding of the threat of the problem and the need for creative solutions. So what I would like to focus on, and is illustrated metaphorically in this little video you're about to see, is the interaction between a bacteria that has spread itself deeper in the body than it should be, and an immune cell, like here, this neutrophil in the bloodstream, that has found the bacteria spreading where it shouldn't be. Now, the job of the immune cell is to, oops, that should be moving. Too bad. Let me try again. Let's just see if this will work. Nope, I'm afraid the video is not working for me. But I will tell you what happened, because I'm a very good storyteller. The bacteria runs around, and the neutrophil chases it, ultimately capturing it. And it's emblematic of an evolutionary dynamic that is going on throughout the eons. The bacteria wants to stay alive, and we want to eliminate it. The problem is we're dividing every 30 years, 40 years if you waste a lot of time in medical school like me. They're dividing every 20 minutes, And that means that for every element of our innate immune system that has evolved, the bacteria have the opportunity to counter that mechanism with their own resistance. They develop an innate immunity to our innate immunity. And that is essentially the definition of a bona fide bacterial pathogen. Staph aureus is able to cause disease in humans because it has evolved resistance to every aspect of our innate immune system. As you can see in this video, it blocks the chemokines that recruit white blood cells to the site of infection. It blocks the deposition and activation of complement. It resists oxidant-based host defenses. It can degrade your antimicrobial peptides. It has the famous protein A that binds your antibodies on backwards so they don't work. It steals proteins from your circulation and hides underneath them. It produces toxins that can kill the white blood cell before the white blood cell kills it. And for all these reasons, it's a very formidable pathogen. It would never cause disease if it was resistant to complement and resistant to antibodies, but exquisitely sensitive to antimicrobial peptides. And that looks very, very scary when you see a pathogen like this, and I could draw a similar graphic for all the leading pathogens. But the good news is that our innate immune system is multifaceted and complementary. And if you are able to discover and understand the molecular mechanism by which the organism is achieving resistance to one aspect of this innate immune system, and you go in and strip the bacteria of that resistance mechanism, you can resensitize it to innate immunity, and that offers opportunities for therapy. So that's basically the theme of this first lecture today, to look to alternatives to classical antibiotics. You know what a classical antibiotic is. It's a chemical that kills or inhibits the growth of bacteria that may be useful in treatment of multi-drug resistant pathogens or in producing better outcomes in serious infections with any pathogen. And this will come in a few different ways as I give you basically vignettes from our research. First is that if you discover a virulence factor, it itself can become the target for therapy. Rather than trying to kill the whole bacteria, you could target the virulence factor. You're not trying to kill the bacterium in that sense. What you're trying to do is disarm it, to render it harmless, to make it no different than all the good citizen bacteria in your body by stripping away its virulence and resensitizing it to our innate immune system. And one huge advantage of this approach is that it can be very specific for the virulence factor and the pathogen that expresses it. Current antibiotic therapy, which is biased towards very broad spectrum agents, does whole-scale damage to our normal flora as a byproduct of the treatment of the infection. These therapies ideally could leave the normal flora intact. A second thing is the modulation of innate immunity to treat bacterial infections. If you have a serious bacterial infection, it's not just that the pathogen had certain virulence characteristics. Obviously, also your innate immune system had a bad day. Bacteria are not supposed to spread from your skin or your throat deep inside your body. The innate immune system dropped the ball. We have hundreds of medicines in clinical practice that are designed to turn down, to dial down the activity of the immune system when it is overactive. A patient has arthritis or multiple sclerosis or asthma, and we use aspirin and corticosteroids and nonsteroidal anti-inflammatory and anti-cytokine therapies to dial down the activity to a more normal level, and the patient's symptoms improve. But I would argue if you have staph bacteria spreading in your blood, landing on your heart valve, and you're very, very sick, why don't we use the immune system in those infections? In current treatment of acute infectious disease, we ignore the immune system entirely. And the immune system is very carefully regulated. It has to turn itself on in response to infection or injury that threatens infection, and yet rapidly resolve itself so that there's not too much damage to the body during the immune response and inflammation incurred by the infection. That means the immune system is a series of brakes and accelerators working together. We have been all over the brakes as a pharmaceutical industry and as a scientific community. Let's look at the accelerators to help treat these difficult infections. And it's interesting, as you'll see, that often you can repurpose existing drugs that are used in other aspects of medicine to show these purposes in the context of the host-pathogen interaction. And finally, I'm going to talk about the synergy between pharmaceutical and endogenous antibiotics. There's extensive research trying to figure out which two pharmaceutical antibiotics work best together, but no one studies how the pharmaceutical antibiotics interact with the endogenous antibiotics, and I think that's a very important area. So as a molecular microbiologist, our tools are based around the concept that my medical school professor Stanley Falco, who won the Lasker Award recently, coined, known as molecular Koch's postulates, trying to figure out the genes that are necessary and sufficient for a virulence phenotype. And this involves cloning genes and mutagenizing genes, trying to create bacterial reagents, living reagents that differ from the parent strain only by the loss or gain of a single gene. And these can then be used to test hypotheses about the role of the candidate virulence factor in disease. And with these living reagents, we can do experiments in the laboratory to understand how the phenotype and behavior of the bacteria has changed. And then we can move into the laboratory for ex vivo studies first. You draw some blood from the principal investigator and purify the white blood cells and mix them with the bacteria and see whether there's any difference in the way that your mutant bacteria interact with the white blood cells. And if it looks like you are on the track of a virulence factor, then you can move to an animal model. And we've used many animal models in our lab, rabbits and rats and mice and fish and flies and worms. But I'm going to focus mostly on mice because of the power of mouse genetics to reveal the interaction of specific virulence factors with specific innate immune mechanisms. And then if everything is looking interesting, like you may have discovered a virulence mechanism, you could ask, could it be a target for therapy? What if you prevented the bacteria from making that virulence factor or somehow neutralized it after it was made? Would that benefit the patient by reducing the virulence of the pathogen? So I'll give you first an older example in the lab that we still work on. Staph is a golden bacteria. Aureus is Latin for golden. And it was known how staph made itself golden for many years. Chemists in Germany had figured out this pathway leading to the pigmented molecules staphyloxanthin and diaponeurosporine, which are yellow and orange in color. We asked a different question in my lab. We asked, why is it golden? In other words, why does it bother to be golden? It's just a prokaryote. There's no fashion sense or anything. There must be some benefit to the organism to go to all the trouble to make these complicated molecules. And if you look at the structure of the pigment, it resembles familiar pigmented molecules like beta-carotene from carrots, or the other carotenoids that are present in colorful fruits and vegetables that I believe in India your mothers also encourage you to eat these foods to prevent against, be healthy and prevent against aging and cancer, etc., because of their well-known antioxidant properties. Well, remember that one of the ways that white blood cells kill bacteria is to generate oxidants. Your white blood cells generate peroxide and bleach, the same things that you might use to sterilize a kitchen counter. And it's very effective at killing most bacteria. But Staph aureus is the #1 pathogen, and it causes infections even in previously healthy individuals. So it must have a relative resistance to this leading host defense. And here we saw the similarity in the structure of the pigment to known antioxidant molecules. And we made the hypothesis that maybe the bacteria was decorating itself with a golden shield against the antioxidant effect of the immune system. And that was an easy hypothesis to test. We generated a knockout bacteria that no longer made the pigment. And we could even introduce the yellow pigment into a normally white bacteria of a different species, here a streptococcus, and make it golden. So we had basically a streptococcus aureus. So you have loss of function and gain of function living reagents to test your hypotheses. First, in a test tube, we're just exposing them to oxidants. Singlet oxygen, oxygen is the most potent oxidant made by human neutrophils. And you can see that losing the pigment, the staph became much more susceptible to oxidants, whereas gaining the pigment, the strep became more resistant to oxidants. And he looked in normal patients' blood, losing the pigment made the staph more susceptible to killing by the immune cells in the blood. But if you took blood from a patient who has a disease known as chronic granulomatous disease, where they aren't able to make the oxidants because of a mutation in the NADPH oxidase, there is no longer an advantage to being pigmented. They're not very good at killing bacteria, but it's only when the host is making oxidants that the pigment provides an advantage. And this could also be proven in a mouse. In a normal mouse that's shaved on its back, the wild-type golden staph can cause an infection or an abscess, but the mutant failed to do so. Whereas in a mouse that has chronic granulomatous disease, they both can cause infection because there's no oxidant defenses. And then the strep that gained the pigment became able to produce a larger lesion. So this pigment was both necessary and sufficient to contribute to virulence, which leads to the question, if the pigment is contributing to the virulence, could it be a target for therapy? What if you prevented the bacteria from becoming golden? Would they be easier to treat? Would you have better outcomes? So now you come to the drug companies with this idea. Drug companies already mentioned in my inaugural comments are not very interested in antibiotics because they don't view them as very profitable. And then you come to them with this idea, well, I'm not even going to have any effect on the growth of the bacteria. I'm just going to change its color, and they're slamming the door in your face. But we got pretty lucky with this because of this interesting coincidence whereby the biochemical pathway by which the bacteria made the pigment had this interesting intersection with the pathway by which humans make cholesterol. And there is a pathway that the drug companies are extraordinarily interested in. And every step of the pathway has been studied, and drug candidates have been advanced to clinical trials. This is downstream of HMG-A-CoA reductase, the target of statins, which I'll return to shortly. But you can see here that in the human cholesterol, squalene is converted into, precursor farnesyl diphosphate is converted into squalene on the pathway to cholesterol, whereas the bacteria take the same precursor and make dehydrosqualene, just differing by a single double bond instead of a single bond. And if you looked at the bacterial enzyme, the dehydrosqualene synthase, it had about 30% amino acid similarity to the human squalene synthase. So we wondered if that similarity might provide an opportunity. And we found that if you took cholesterol drug candidates that had advanced through phase 2 clinical trials as squalene synthase inhibitors, they were able to crystallize right into the active site of the human drug. Of the bacterial enzyme. The human drugs crystallized into the active site. This led to an inhibition of the enzymatic activity, sensitizing the bacteria to oxidants by eliminating the pigment. And the bacteria that had been stripped of their pigment pharmacologically with no effect on their growth became more sensitive to killing in human blood, and in an animal model of infection, systemic staph infection, treating them with this cholesterol drug candidate that prevents the bacteria from becoming golden, had a therapeutic effect in a couple of days that was similar to what you might expect with an antibiotic like vancomycin. So a purely virulence factor-based therapeutic having an activity. I want to give you one more example to illustrate a phenomenon known as neutrophil extracellular traps, which was first introduced on the cover of Science by our colleague Arturo Zitslinski, who is at the Max Planck Institute in Berlin. And this is a new understanding that neutrophils do a lot of their killing extracellularly, not just by phagocytosis. Because when they get to the peripheral tissues, they can undergo a specialized cell death process where they spit out their nucleus, and it unravels in the tissue to make a network that is very sticky and full of antimicrobial compounds, including antimicrobial peptides and histones, which are very cationic and antimicrobial. And within this network, bacteria are trapped and killed. This is basically what pus is. No one is that excited about studying pus because it's, distasteful. But it's very interesting. It's kind of like a quicksand to trap bacteria at the site of infection. So right when this paper came out in Science, we were studying a clone of the other leading human gram-positive pathogen, group A streptococcus, that had disseminated globally to be the leading cause of severe invasive strep infections like toxic shock syndrome and necrotizing fasciitis, the flesh-eating disease. Muppeteer, all of you probably know Kermit the Frog and the Muppets. Jim Henson died in his 50s due to invasive group A streptococcal infection. And a single clone in the last 3 to 4 decades has disseminated across the globe to produce these severe infections. And our lab has been studying the virulence mechanisms of the pathogen. In a proteomic analysis of this particular clone, we discovered a unique protein that was present in this strep, but not in other strep. And it turned out by analysis to be a type 1 DNase. The bacteria was making a DNase, a DNA degrading enzyme. And the cover of Science is telling me the new way that the immune system kills bacteria is to trap them in DNA. And here I have this pathogen that doesn't seem to be getting cleared by the immune system. It's spreading through the body, and it's making a DNase. In fact, it had come in on a bacteriophage. The bacteria got infected with a virus that gave it a new gene and created a superbug. Very similar to what happens with antibiotic resistance. Here, the selective pressure is the innate immune system that it becomes resistant to. Currently, with antibiotic resistance, phage and plasmids often bring resistance to the pharmaceutical antibiotics that we're trying to treat them with. So we decided to study this in the same way, a molecular genetic approach. We took the flesh-eating strep, which made this potent DNase, you see here degrading all the DNA, and knocked out the gene. So it could no longer degrade DNA. And we could also introduce the DNase in normally skin-associated non-virulent strains of strep, and they gained the ability to degrade DNA. Look what happened in the animal model. Losing the DNase made the flesh-eating strain much less virulent, whereas gaining the DNase conferred virulence onto the skin-associated strain. And in pharmacological studies, we showed that an inhibitor of the DNase, G-actin, could completely eliminate the survival advantage of the wild-type strain over the mutant, and this very same inhibitor, when used in an animal model, which is being infected with the flesh-eating strain on both sides, had a marked therapeutic effect by inhibiting the DNase, preserving the nets, and allowing the infection to be controlled and cleared by the neutrophils at the site of infection. Statins. This is going to link the two stories that I talked about. So statins are cholesterol-lowering drugs that are used worldwide by now hundreds of millions of individuals. They inhibit HMG-CoA reductase. They're very good at lowering LDL cholesterol. In America, there's probably almost 40 million users now. With the new recommendations by the American Cardiology Association, probably 100 million Americans should be taking statins. I became interested in data that was emerging from the clinics, from my experience interacting with other physicians and physician scientists, which showed that patients who happened to be on statins were doing better than patients of similar age and disease state that were matched controls with a variety of different infections like bacteremia, sepsis, pneumonia. And I knew that Staph aureus was one of the leading causes of these infections. And there was a prevailing theory that statins have known anti-inflammatory activities, and maybe these patients, their sepsis wasn't so bad because they had an anti-inflammatory effect. But that didn't make sense to me, because in general, if you give anti-inflammatory or immunosuppressant drugs to individuals, they become more susceptible to infection, not less. So I wondered if it could be something more fundamental in the host-pathogen interaction, such as the interaction between the white blood cells and the bacteria, something so easy to test. We just took different kinds of white blood cells, neutrophils and macrophages from humans and mice, exposed them to statins for a few hours, and then did a bacterial killing assay, and found that they were able to kill bacteria better. And this was all sorts of different kinds of immune cells and all sorts of different kinds of bacteria. Just a few hours exposure to this drug taken by hundreds of millions of Americans. Interestingly, phagocytosis and oxidative burst, consistent with prior literature, were diminished in the statin-treated white blood cells. So this led us to think that it can't be an intracellular killing mechanism. It must be extracellular and therefore related to this new phenomenon of nets. And what we found out is that statin-treated white blood cells, although they do not make a net simply on exposure to statin, if they then see a bacteria, they make nets much more efficiently and robustly. And these nets are full of antimicrobial molecules, like the antimicrobial peptides. And this was any statin that worked. In fact, if we used RNA inhibition to knock down the pharmacological target of statins, HMG-CoA reductase, we also increased net production and increased bacterial killing. In fact, macrophages will make Mets, I guess you call them. It's kind of a baseball term in America. If you stimulate them with statins and expose them to bacteria. Could this be useful? Well, we went to animal model and we treated mice with the doses of statins that would be given in humans, and then pulled white blood cells out of those animals and tested their ability to make nets and kill bacteria, and they were superior to the control animals. We then infected statin-treated animals versus control animals, and they had better bacterial clearance in this model of staphylococcal pneumonia associated with increased production of statins. So, a drug taken by hundreds of millions of Americans fundamentally alters the way your white blood cells behave when they interact with a pathogen. And in the case of Staph aureus, the clinical data and the experimental data say it could be beneficial. And this seems like a ready intervention that people could use. And we've continued to explore drugs approved by the FDA for these behaviors. Here is a dramatic example we recently published. Tamoxifen, one of the mainstay drugs in the treatment and prevention of breast cancer, has an activity to enhance the production of neutrophil extracellular traps. And it does so not by interacting through estrogen receptors, which is a mechanism for anti-cancer effect, but by another known effect of tamoxifen to block this enzyme, glucosylceramide synthase, to allow accumulation of ceramide in the cell, which activates PKC zeta pathway known to stimulate netosis. And in experiments treating mice with tamoxifen, a breast cancer drug, and no antibiotics, provided protective activity and better clearance of bacterial infection than untreated animals. Very interesting that these other drugs could be explored in that way. In our collaboration here at Amrita University and with Professor Perry from UC Riverside, we have been looking at interesting molecules that are natural products derived from, I came from Cullum this morning along the highway, and I saw the signs for the cashew factory and have seen the trees on the property where I'm staying. The cashew nut shell has a compound, anacardic acid, which is known to have anti-staphylococcal activity. But because of its structural similarity to a known stimulator of neutrophil activity, known as PMA, we explored further, and in fact, we have discovered that the administration of anacardic acid is able to stimulate enhanced reactive oxygen species generation in neutrophils and better production of neutrophil extracellular traps. They modify HIF so that it interacts with a ubiquitin ligase known as von Hippel-Lindau factor. The transcription factor is destroyed then by the proteasome. So if you measured HIF levels or activity in your white blood cells in your bloodstream where oxygen is high, you would detect 0, even though it's continually being made. But when oxygen is low, HIF is no longer degraded because the prolyl hydroxylases don't work. HIF is then able to go to the nucleus, and it stimulates a lot of genes to turn on. And what do those genes control? Well, if you don't have any oxygen, your cell still needs to generate ATP, and it does that through glycolysis. And 32 out of the 34 enzymes in glycolysis are directly regulated by HIF. So it's the master regulation of glycolysis. It also controls an interesting hormone known as EPO, or erythropoietin. And many of you probably know EPO because of its use as a drug in sports cheating in the Tour de France to boost your blood carrying capacity and oxygen. But it's also a very important drug in human medicine for people who have anemia. Like chronic renal failure patients have anemia, and they want higher blood levels, and they're treated with EPO. So the pharmaceutical industry has been very interested in discovering other mechanisms to boost the production of EPO in the body. So since HIF controls EPO, they have developed inhibitors of the HIF degradation pathway, so that HIF is not degraded, it accumulates, and your body makes more EPO. And in fact, these are in advanced clinical trials, phase 3 clinical trials, thousands of patients, multiple companies, and they're soon be on the market as anemia drugs, prolyl hydroxylase inhibitors that boost HIF. So just remember that. There are drugs that boost HIF by inhibiting the prolyl hydroxylases. So why am I interested in HIF? Well, in collaboration with Randy Johnson, who was at my university but now is in Cambridge, England, he was the first to be able to knock out HIF in specific cell lineages in the mice for further study. You can't knock out HIF in the whole animal, that's embryonic lethal because it's so important. But you can knock out HIF just in the white blood cells through a system known as the Krelok system to delete HIF under the control of the lysem promoter, only expressed in these white blood cells. And it's interesting that losing HIF, the mouse develops normally under standard housing conditions. Its white blood cells can phagocytose bacteria, but they can't kill bacteria. So there's something wrong with their immune system. And when we looked at it more closely, they were unable to make those antimicrobial peptides that I described just a few moments ago. So HIF is the master regulator of the production of these antimicrobial peptides, and there is a HIF promoter in both the human and the murine cathelicidin. A few other activities like granule proteases and tumor necrosis factor and nitric oxide are also controlled by HIF. Knock out HIF, the activity goes down. Knock out VHL in the HIF degradation pathway, HIF accumulates, the activity goes up. In fact, infection or exposure to bacteria is a stronger stimulus for HIF accumulation than is low oxygen. I don't think I'm going to be able to get the name of this transcription factor changed, but it's an integral part of your innate immune system. And if you knock out HIF in the animal, in their white blood cells, they are unable to control infection. So why would you regulate your immune cell activity with HIF? Well, it's actually a pretty good system if you think about it. You don't want to be inflamed all the time, although I think we all know some people who are. You would like your white blood cells to be quiescent, at rest, and as they circulate through the bloodstream with high oxygen, that is what they are. But when they get a signal to go to the site of infection, they immediately encounter a steeply declining oxygen gradient. At peripheral foci of infection are either severely hypoxic or anoxic. The bacteria like staph and strep, they love it. They grow better with no oxygen. But your white blood cells have to go into this environment holding their breath and fight the pathogen. And the way that they've done that is to generate all their energy through glycolysis. And because they generate all their energy through glycolysis, they are very strongly regulated by HIF. So why is that interesting? Well, if you look at the white blood cells from mice that have elevated HIF, because we have genetically eliminated the degradation pathway, those white blood cells kill bacteria better than normal white blood cells, which is to say that your white blood cells aren't as good as they could be because you're trying to regulate when you turn them on and off. But what if you have a severe antibiotic resistance, staph or Pseudomonas infection? Wouldn't you want your white blood cells to be a little stronger, especially if the antibiotics have a risk of failure? Well, that is what we explored. So where did we go? Well, remember the anemia drugs, the drugs that boost HIF that are being developed to increase EPO levels in the patient. We wondered whether they could also boost the antimicrobial performance of the white blood cells. And what we found is that if you treat macrophages with these HIF boosting drugs, you can elicit the macrophages to kill the bacteria very effectively, because the macrophage is able to make dozens of different antimicrobial compounds. And in fact, it worked better than vancomycin at 2 times the MIC, and we had a nice therapeutic effect in this animal model of MRSA infection, repurposing anemia drugs for the use in infection. And we've gone on to show that we can treat gram-negative infections, such as the E. coli urinary tract infection, in which antimicrobial peptides are also important by boosting HIF. And perhaps our most dramatic result is in a paper we're about to submit, the lethal bioterror threat, anthrax, as part of its mechanism of action, suppresses HIF production. And we discovered that you can pharmacologically reverse this suppression and have dramatic effects, saving mice from a lethal challenge of anthrax through a host-directed therapy with no direct antimicrobial activity. So just this whole area, I think, is very interesting. And another thing I just want to throw out there, this is a paper we're writing up also, is that there's a whole other group of cells in your bloodstream that have been entirely neglected for their role in innate immunity, but they are very important, and they are platelets. In fact, platelets have very strong antimicrobial activity against Staph aureus. And in order to survive in the bloodstream, Staph aureus has to eliminate the platelets by producing a toxin known as the alpha toxin, which lyses the platelets. And it's a true arms race. The platelet will kill the staph if the staph doesn't kill the platelet first. And we have found that a common drug prescribed for millions of individuals known as ticagrelor, very similar to a drug known as Plavix, which is a P2Y12 inhibitor used as an antiplatelet drug for patients who've had myocardial infarction, heart attacks, or stroke, is able to protect the platelet from this toxin and reverses the outcome in infection, and has a very strong protective effect against staphylococcal bacteremia. And we have some human clinical correlative data that are consistent with this finding. So, host-directed therapies. All right, so I have the last topic, which is about synergy, and it's going to take me into the world of gram-negatives, where I had never gone before, and has been a little bit intimidating. But it starts with a quiz that I don't really expect anybody to answer, because I couldn't answer before I found the photos myself. But these are 2 notable individuals in microbiology and medical history. anybody have any idea who these individuals are? Well, the man on the left was the former chairman of microbiology at Harvard for decades. And the man on the right was the first African-American professor at Harvard University. They are John Howard Mueller and William Augustus Hinton. And I have William Hinton here only because he's the father of the true heroine of the story, Jane Hinton, whose picture I can find nowhere. Was a very interesting woman in her own right, the first African-American female veterinarian in America. After she finished her DVM degree, Jane Hinton did a postdoc with John Mueller. And they had this paper, small paper, little subspecialty journal in clinical microbiology. And the paper had a goal of improving bacteriologic media so that you could recover more bacteria from clinical specimens. And in particular, they were worried about gonococci and meningococci, which are very fastidious bacteria. They're very picky eaters, so to say, and they need very rich media. And they came across this concoction of beef infusion and starch and agar, and later it was modified by the addition of salts to improve recovery of Pseudomonas, and it's known as Mueller-Hinton broth. Well, Mueller-Hinton broth somehow, over the decades, has now become the single broth that is used in the clinical laboratory to determine the MIC and MBC, the antibiotic potency of all the drugs we use for most of the common bacteria. So the original purpose of the drug, of the media, was to enhance recovery of bacteria. But now it is being used to pick the antibiotic to treat the patient with. A single test in a single arbitrary media that just came about in this kind of random way now defines pharmacotherapy for the entire specialty of acute bacterial infection disease. Really interesting. But one thing I want to point out is that this is where the infection is. The infection is in a human, and that human is not made out of beef extract and starch and casein and agar. It is made out of tissue fluids and organs, and importantly, before the patient has ever seen the doctor, their infection is already being treated by dozens of antibiotics, all these antimicrobial peptides that your body makes. And our current testing paradigm for infectious disease is completely agnostic of this. We don't include the immune system. Here's an example of a patient with methicillin-resistant Staph aureus of the nightmare variety. 21 days of positive cultures, the organism is becoming resistant to vancomycin and daptomycin. Endocarditis. Because of some experiments that we had been doing in the lab, identifying potential synergies between beta-lactam antibiotics and daptomycin, which was being administered fruitlessly in this patient, we introduced nafcillin. So nafcillin is basically methicillin. This is methicillin-resistant Staphylococcus aureus. Nafcillin has no direct activity against this pathogen. But the next day the culture was clear, and this patient was cured. In fact, we published a series of 10 other patients with similar outcomes. What was happening? When you gave a little bit of nafcillin to the patient, all of a sudden, cationic antimicrobials, including the drug daptomycin, but importantly, the innate antimicrobial peptides that your body is already making, begin to target the bacteria and have their killing activity. And you can see that tiny doses of nafcillin, well below the MIC, sensitize methicillin-resistant Staph aureus to killing by whole blood neutrophils, keratinocytes, which make the antimicrobial peptides, and in fact, a drug with no direct activity against the pathogen, because of its synergy with innate immunity, has a demonstrable therapeutic effect in vivo. And these papers that we've published have led to a change in the official guidelines in America advocating the reintroduction of nafcillin for MRSA in combination with daptomycin for recalcitrant infections. We don't know why it shouldn't be used at the start, but we'll take this first step along the pathway. Similar results we've published for vancomycin-resistant enterococcus, reintroducing ampicillin, even though there's no activity in a test tube, and having dramatic clearance. Okay, so now the gram-negative story and the last story I have to tell. So you all are aware of these multi-drug resistant gram-negative pathogens such as Pseudomonas, Acinetobacter, Klebsiella, the NDM1 producing E. coli, that are resistant to all the advanced antibiotics. And it's a huge threat with high resistance rates. Here are the isolates that we're using in this set of experiments I'm about to describe. Resistant to everything except colistin. But remember, the resistance is based on this arbitrary test in a media which does not equal the patient. And we wondered, what if you just did the test in a more physiologic conditions? First, by just doing the testing instead of the bacteriologic media, how about doing the testing in tissue culture media? I mean, tissue culture media is designed to simulate, human tissue fluid concentrations and support the healthy growth of cells. Well, here's the MIC, highly resistant, of the most common drug used in America, azithromycin. Never recommended for gram-negative bacterial infections. Never prescribed for these serious infections, which have maybe 30% mortality rates. Why? Because it has no activity in the standard testing. But if you do the testing in tissue culture media, you have very potent, favorable MICs. So what is the MIC? Is it this? Because that's the way we've always done it? Or is it this? To go beyond that, here's what you see. Multi-drug resistant Klebsiella pneumonia, multi-drug resistant Acinetobacter, no activity of the drug in standard bacteriologic media, in the presence of tissue culture media, bactericidal clearance within 24 hours. But here's the more dramatic result. There is massive synergy of these antimicrobial peptides with azithromycin. At 1/4 the MIC of azithromycin and your endogenous antimicrobial peptide, put the two drugs together, multi-log bactericidal synergy. And why is that? Because when you have the physiologic concentration of antimicrobial peptide present at every focus of infection, the cell wall is weakened enough to allow azithromycin to get inside the cell and then just do what it's always done, inhibit protein translation through the ribosome. Here's the standard testing. That's what's happening to your loved ones when they get an infection in the hospital. This is the condition in your body. And a totally effective antibiotic is discounted because of the narrowness of our testing paradigm. Would this work in vivo? Here we are treating mice infected with these multi-drug resistant bacteria that I showed you in that MIC table, and giving them 1 dose of azithromycin. And you see 2 to 3 log reduction in bacterial counts, 99 to 99.9% clearance with a single dose of azithromycin, associated with less inflammation in the lung, and at a higher dose, which produces lethality, saving the animals with 2 doses of azithromycin. And we've gone on to do experiments with Klebsiella pneumoniae, Pseudomonas, a separate paper on Stenotrophomonas, even more esoteric multi-drug resistant bacteria now. The synergy also exists with colistin, and easily demonstrable even in the standard tissue culture media. Potent synergy between azithromycin and colistin that could be used. So I'm going to wrap up there. We're in San Diego. We got to move into a brand new building. We have a lot of interesting people in the lab and frequent visitors from all over the world. San Diego has a lot of biomedical conferences, a lot of biotechnology industry, which may bring all of you there at some point, and we hope you come and visit us. But more importantly, we hope that you're inspired to think outside the box about antibiotic therapy and recognize now that we can take inspiration from these proofs of principle, that there's more to antibiotics than just chemicals in a test tube. Think about cancer, and this will be my last point. What were the first cancer drugs? They were also chemicals that were screened in a test tube for their ability to kill dividing cells. Mustard and cyclophosphamide. And what happens to the patient when they get those drugs? Well, their cancer is shrunk, but in addition, they lose their hair, they lose their mucous membranes, their bone marrow is suppressed, and a lot of medical problems. But there is a revolution in cancer therapy that has come in the last decade. About 10 years ago or so, a drug was introduced called Gleevec, which is a targeted drug that inhibits the kinase that is mutated in chronic myelogenous leukemia and has cured a lot of chronic myelogenous leukemia. That is exactly analogous to treating a virulence factor of a pathogen. It's targeted to the problem in the cell. It's just a bacterial cell instead of the human cell. Our ex-president, Jimmy Carter, 92 years old, was diagnosed with malignant melanoma to the brain. That is a death sentence in a 19-year-old 6 years ago, in months. He gets a new drug that is a checkpoint inhibitor that unlocks the ability of his T cells to attack the cancer. 4 months later, he declares himself cancer-free. He celebrated another birthday. I think he's boycotting the inauguration, but he is well and doing his humanitarian missions. That is exactly analogous to a drug that would treat the macrophage or the neutrophil and boost their ability to kill disease. We have become complacent as infectious disease physicians and researchers because antibiotics work so well for so long. We treated more patients than all the other drug classes combined successfully. But our luck is running out. And there's these multi-drug resistant strains. And the golden era of antibiotics is coming to a close. We need to be inspired by the cancer doctors. They never had the luxury to become complacent. Every family is affected by cancer. Many cancers have terrible outcomes. They never stopped thinking. They never stopped discovering. And they've had huge advances. And all we need to do is follow that kind of thinking. If you are diagnosed with a cancer that has a 30% 5-year mortality, what happens? A whole team gets together in a room, tumor board, radiologist, cancer doctors, surgeons, clinical trialists, pharmacists. They repeatedly discuss your case to find the optimal therapy and options, usually including immunotherapy as a forefront now. What if you have a serious infection like Acinetobacter and 2 positive blood cultures? You have 30% mortality too, but not 5 years. This hospitalization. What is happening to you? Probably a junior resident is looking at a series of R and S's on a computer that were calculated in Mueller-Hinton broth in a clinical lab and making their best guess as to what to use among the S's, if there are any. I think we can do better than that in the field, so I hope you all join us, and thank you for your attention. I know I ran a little long, but... Questions now, or? Open to the audience. Okay. I'm opening it up to the audience for any questions. There's no question that is too simple or naive. If you could really think about it, the genesis of all the different projects I talked about was very simple questions. Why is the staph golden? You know, why does an oxygen regulating factor control immune system activation? Why do we do all our testing in a single media that has nothing to do with the body? These are very simple questions. And then if you step back and think about it, you can come up with experiments, right? And who knows what you'll find. Yes. It's coming.
Professor, very fascinating presentation. Thank you. As a pharmaceutical scientist, I have 2 or 3 questions.
Please.
You mentioned about non-antibiotic answers to bacterial resistance and all infections in general. One, of course, is azithromycin. Then you mentioned about tamoxifen. And the third was...
Statins.
which one?
Statin drugs.
Statins,
Yeah.
these 3. Now, what happens is that if you look at statins, there's recently a shadow on statins, particularly a shadow on the role of cholesterol in the outcomes of cardiovascular events.
Yes.
And that debate is going on.
Very much so.
But in any case, since statins have HMG-CoA reductase activity, and it leads to cholesterol synthesis, what is the role of cholesterol itself?
Yes, that's a very good question. Yes. So...
Can I finish or one by one again?
I will answer that question and then invite you to have a follow-up question.
Okay.
So it is interesting that there, in fact, is a contribution of cholesterol itself to the phenotypes we're observing. If you add exogenous cholesterol, you can have the opposite effect to suppress the activity of the white blood cell. And in fact, HDL cholesterol has a very important role in innate immunity that we are also defining, which involves neutralization of certain bacterial toxins. A number of bacterial toxins that cause the dysfunction of critical components of innate immune system activity in white blood cells have a cholesterol dependency in their initial interaction with the cell membrane. And by reducing cholesterol, the targeting of the immune cell through its membrane is diminished. We also believe there are roles of lipid raft signaling, and that certain byproducts of cholesterol that are even further downstream, but nevertheless inhibited by statin treatment, interact with nuclear receptors that have anti-inflammatory effects on the cell. So by reducing these ligands for the anti-inflammatory nuclear receptors, you unlock the immune activation potential of the white blood cell. And that is consistent with the effect that we saw that not only the statin drug, but the pharmacological inhibition of HMG-CoA reductase, or the genetic inhibition, led to the same phenotypes and therapeutic benefits.
Well, does it mean that other sources of cholesterol biosynthesis, including some natural products like gugulipid, I don't know whether
Yes.
you heard of gugulipid. It
I have
comes from
not, but
it
I'm
comes from a
very
tree called
intrigued.
Commiphora mucul.
I see.
It is an anti-lipidemic drug.
Yes.
So should
Could
we look
also
at
have
this-
benefits,
Since
yeah.
statins have other problems, should we look at other agents which are able
I
to,
think
able
that's
to corner
very
lipid profile?
very likely that agents that have similar potency in cholesterol lowering
And
would
triglycerides.
also have immune boosting properties. I agree with you. I would also point out, though, that remember, a lot of the downside of statins that come from, muscle inflammation and other adverse effects that patients have. Remember, we're only talking about treating the patient for a very short period of time as an adjunct to their serious infection. And the inflammation caused by the serious infection that is under control is often causing a lot more harm and dysregulation to the host than the short-term administration of a therapy. But by and large, although statins have a number of problems, there is a large body of evidence of a benefit for a huge, segment of the population who are lucky enough not to experience the adverse effects.
So would you be able to look at gugulipid, for example?
I would love to
Yeah,
receive
maybe let's
a
release.
sample of that in the mail
Yeah.
and tell you exactly the efficacy.
Yeah. Second thing is about tamoxifen.
Yes,
Tamoxifen is a selective estrogen modulator.
yes,
No, it's a SERM.
yes,
So, and there are SERM which actually inhibits the production of estrogen. So
yes,
the role of estrogen, as well as other drugs like letrozole, for example, which is an aromatase inhibitor, which also inhibits the production of estrogen.
yes.
Role of estrogen in women for the case of infections, for example, of various types.
Right. So I think it is very interesting area of research. We believe that the pharmacological, this particular narrow pharmacological activity of tamoxifen that we studied is independent of its role as a selective estrogen modulator, because we did multiple inhibitors and agonists of the estrogen receptor pathway without having the similar effects, and yet by targeting glucosylceramide synthase, which was a known off-target effect of tamoxifen, we were able to reproduce the phenotypes. Recently, raloxifene, which is a different type of inhibitor, actually has exactly the opposite effect on nets, and we recently published that paper,
So that's
which means that there is a role of estrogen receptor signaling in controlling these phenotypes that maybe merits exploration and maybe could be pushed in both directions, and could also explain, for example, in pregnancy, women are much more susceptible to a wide variety of infections. In general, that has been felt to be related to T-cell immunity. You have a living person in your body, the fetus, that can be at least 50% genetically dissimilar, and we know from surrogate pregnancies can be 100% genetically dissimilar, and fetus tolerates mother and mother tolerates fetus by suppressing immune recognition, and that leads to increased susceptibility to infection. But the high hormonal levels of estrogens and progesterone are also very likely to have effects that are worth studying, both on mucosal surfaces and on systemic immunity. I mean, if anything changes cell biology significantly, you look at a Staph aureus fighting a macrophage. This is a death match, and every system is brought into play. In order for the macrophage to win, not only does it have to produce antimicrobials, but it has to generate energy, repair its membranes, handle oxidative damage, handle damaged subcellular organelles, autophagy, ER stress response. These are all part of innate immunity. Dr. Banerjee is going to illustrate that dramatically with his research on autophagy shortly. And so this means that everything in cell biology, all the reasons that we have drugs in human medicine are because they take non-functioning cells or organ systems and alter their physiology or metabolism in a way that restores health to the patient. If you study them in a host-pathogen interaction, they are very likely to have an effect. Some of these might be beneficial, some of these might be detrimental, but I think it's very important to go back through all the approved drugs and try to find new therapies. You can have an immediate impact on clinical care.
Repurposing.
Repurposing.
The last one is
Yes.
about azithromycin.
Yes.
Azithromycin is a drug of choice for upper respiratory infections, including staph, including strep.
Yes.
Now, you mentioned something about tissue media against conventional media. I didn't quite
Yes.
understand the difference as to how you get totally different results with respect to.
Well, I don't know.
either
I think
MIC
that's
or-
like, all of us who are scientists in the laboratory know that when you change the experimental conditions, you can get different results.
But what's the
It wasn't
significance?
something that I predicted would necessarily happen, but we are desperate, in these patients who apparently have no drug to treat
But
them,
what's
to
the
really try to
significance?
understand it better.
What's the significance from the clinical point of view?
The significance is that the testing is only done in bacteriologic media, and the bacteriologic media inhibits an activity that we can easily demonstrate in the context of white blood cells in the whole animal. So that's the significance, that we have a single test dictating our approach to a problem that is much more complex. Yeah. Yes.
I think farnesyl pyrophosphate inhibitor can have some role in this. You're talking about statins.
Yes.
And that's in the farnesyl comes much later.
Yes, no, I agree. So we know that, for example, that when we looked for drugs that, initially when we were trying to inhibit the pigment of the staph, we were thinking in kind of the old mold that we would want a drug that was most selective for the bacteria to avoid any chance of effects on the host. But then when we discovered this role of the cholesterol pathway in nets, we reapproached our analysis looking at squalene synthase inhibitors that had dual activity against both the human and the bacterial enzymes, and were able to show up to 40-fold greater activity in our assays by presumably sensitizing the bacteria to oxidants while simultaneously increasing the killing activity of the immune cell. And I would predict that it is per se anything on the cholesterol pathway and not on granulation or other branches that are downstream of squalene. So anything along that pathway would have a similar effect in supporting innate immunity and is worth exploring.
One of the medicinal plant contains isoramnitin, which is supposed to have this activity.
Well, that would be another great. So you already have 2 sets of experiments, and I know just who can do these experiments, too.
So,
Hi, Ajit. Yeah.
so inspirational presentation. So, as a follow-up of Dr. Nayer's question, the last question indeed, the profile of sensitivity or resistance of an organism is quite different in a bacteriological media and a tissue culture. In that particular background, are we overestimating or underestimating the antibiotic resistance as such?
Yes, no, I think every example is possible. I mean, we have been biased to try to identify examples of underestimation because we're looking for solutions for desperate patients. But I can tell you, for example, we did research to show that bacteriostatic antibiotics, of which there are many, for example, tetracycline or chloramphenicol or even erythromycin, Bactrim against certain concentrations against different organisms, are bacteriostatic. And when you suppress the growth of a bacteria, although that may be beneficial for the host, it actually antagonizes the ability of the natural antibiotics to kill the bacteria. Because the natural antibiotics are waiting for the bacteria to divide. Because in order to divide, to be one bacteria and become two, you have to weaken your cell membrane enough to split into two. And it's that moment of vulnerability that most of the killing goes on. This is also true actually for beta-lactam antibiotics, that penicillin is most active on logarithmic phase bacteria and loses activity. It's something called the eagle effect on stationary phase bacteria. So bacteriostatic antibiotics have a role in human medicine, but it's despite antagonizing your innate immune system. And the superiority of bactericidal antibiotics is in part related to not interfering with the innate immune system and working side by side with it. So I believe if you systematically looked at all the antibiotics and did it with physiologic conditions, and you had a rank order in the bacteriologic media and a rank order under the physiologic conditions, that they would be different. And there would be examples of drugs moving up and drugs moving down depending on the pathogen. It's just something that needs to be explored. And that's exactly what we're doing with, for example, with Roche. We have a collaboration where we're screening their entire library, 800,000 compound library of chemicals, but instead of screening for their ability to directly kill a bacteria in bacteriologic media, which has been done for 40 years, we're screening in a little mini immune system where we have white blood cells and serum under physiologic conditions and the bacteria. So now we ask, under those conditions, are there any drug candidates that kill the bacteria? And then we figure out how it worked. It could have been a direct antibiotic, but it theoretically could have been a drug that sensitized the pathogen to innate immunity, that synergized with innate immunity, or in fact worked through the white blood cell to increase its killing activity. And it's pretty simple to deconvolute the assay and determine what the mechanism is. You just eliminate the white blood cell and eliminate the serum and repeat it until you understand the mechanism. But it's not that hard. Let me ask you this. Let's say you were really sick in the hospital with a drug-resistant infection, and I came to you as your doctor, and I said, we can do the testing of the different available antibiotics. Would you like me to do the testing in the bacteriologic media or in your own blood where the infection is? It's not harder to do it in the blood than in the bacteriologic media. It's the same test. So which information would you like if I can only give you one test? I mean, to me, it's just a simple kind of logical thing. We have to deal with a bureaucracy. You know, this testing has been extraordinarily valuable. There's value in the testing. The testing you get in every lab is the same. There's tremendous value in that. You can follow epidemics of infectious disease. But we now have bacteria that are resistant to all the antibiotics. And you can't just say, no further testing. You have to think about what else can you do to analyze the patient. And doing the testing under different conditions is an example. Because the only other alternative, and in fact what a lot of people do, is they do experiment on the patient with no further information. They just start throwing more antibiotics at the patient and then see if they do better or not. But that doesn't seem very logical or information driven, so we'll see if we can do better. Pippin, Professor.
We could just keep on asking you questions, and
I'm around
we
for
stuck.
both it is.
So in the interest of time, we'll go on to a short tea break and then come back in for Anirban's talk. And
Excellent. I
Thank you
look
so
forward
much
to that
for
myself. You're welcome.
To Dr. Nizet.
Wow. Beautiful mirror.
Special thing from Kerala.
From
Metallic
Kerala?
mirror.
Yes, it's a beautiful, beautiful mirror that will be right on my desk in my office and always remind me of this visit. Thank you so much.
You're making things difficult for us. There are too many problems to solve.
Yeah, well, it's a challenge. Thank you very much. Thank you, everyone, for your attention. Enjoy the break.
Thank you, Dr. Nizet. Thank you, Dr. Banerjee. We now take a short tea break, and the session will resume after 15 minutes. Thank you.